Hello everybody, my name is Hannah and today I'm going to be teaching you guys about the cell cycle. You might be wondering, why is the cell cycle important? Well, the cell cycle is important for regulating cell division. It enables organisms to grow, develop, repair damaged tissues, and maintain appropriate cell counts by controlling the timing and frequency of cell replication. This regulation helps prevent uncontrolled growth, which can result in various medical issues. Essentially, the cell cycle functions as a quality control mechanism for cellular reproduction, checking for DNA errors before the division process. Now that we understand why the cell cycle is important, let's examine the phases and talk about how they work. Let me introduce you to interphase. During interphase, a cell increases in size, replicates its DNA, and prepares for cell division by duplicating organelles and synthesizing essential proteins. This stage, which constitutes the majority of the cell life cycle, involves the cell performing its normal functions while gearing up for the mitosis phase when it will divide. After interphase comes our next phase, prophase. During prophase, the chromatin within the nucleus condenses into visible chromosomes, and the nuclear membrane disintegrates. Simultaneously, the spindle asparatus begins to form, with centromes migrating the opposite poles of the cell in preparation for the separation of the replicated chromosomes in two distinct daughter cells. This phase marks the cell's preparation for the division as it organizes the chromosomes into defined structures. Now we know about prophase, let's move on to metaphase. During metaphase, all chromosomes in a cell align along the equatorial plane, known as the metaphase plate. They become fully condensed and attached to the mitotic spindle fibers, preparing to be separated in two daughter cells. In the subsequent phase, anaphase, this precise alignment is crucial for ensuring that each new cell receives an equal set of chromosomes. After metaphase comes an important phase called anaphase, right here. During anaphase, the sister chromatids of each chromosome separate at the centromere and are pulled apart by spindle fibers which move them towards opposite poles of the cell. This process divides the replicated genetic material into two distinct sets that will eventually form separate daughter cells. It occurs as a result of a breakdown of the proteins that hold the sister chromatids together, enabling their separation and movement to opposite ends of the cell. Anaphase was cool. Now let's learn about the final phase, telophase. Right here. During telophase, the final stage of mitosis, the chromosomes that were separated to the opposite poles of the cell during anaphase begin to decondense. New nuclear membrane forms around each set of chromosomes, resulting in the creation of two distinct nuclei. While the mitotic spindles disassemble, this process essentially reverses the changes that occurred during the phase in the premetaphase, setting the stage for cytokinesis. Okay, now that we have learned all about the phases in the cell cycle, let's talk about our special mention, cytokinesis. Cytokinesis is the process in which the cytoplasm of a single cell divides to form two distinct daughter cells. This occurs through the inward pinching of the cell membrane at the equator, creating a cleavage furrow that separates the newly divided nuclei into individual cells. This mechanism ensures that each daughter cell receives a complete set of organelles and sufficient amount of cytoplasm from the parent cell. This was my video on the cell cycle. Thank you for listening.